So in this lecture, I'd like to continue the discussion of morphospecies by revisiting the Sungai Matan study, where we compared selectively logged forests with undisturbed forests in a nearby site. And this was all based upon the morphospecies concept. And I'd like to explore the question, can we crowdsource the morphospecies concept and involve citizen scientists in this research? Again, we're going to revisit this study that compared commercially logged Bornean rainforest. And let's talk about where we are, just to make sure everybody's on the same page. We're talking about Southeast Asia between the Indo-Chinese continent and Australia. It's a very interesting region for many reasons. One of the most important is because of Alfred Russel Wallace defining a major biogeographic boundary in the region that separated two ancient continents from one another. And we're specifically speaking about the island of Borneo. It's a major center of diversity and endemism, and it lies right on the eastern margin of Wallace's line. It's a major island divided between Malaysia, which has the northern half in Sarawak and Sabah, and then there's Kalimantan, which is the Indonesian portion of the island, and that's where the research was conducted. On the western coast of Borneo, there's a large national park called Gunung Palung. That's where I first went to study as an undergraduate. It's a small, isolated mountain range, and you can see here it's very isolated from the central mountain range of the island. It's a very interesting area. There's a research site there called the Cabang Pantai Research Site. It's been there for many, well over 30 years now. And back at that time, when I was an undergraduate, you took long river trips to get to the research station. We were really very isolated, very basic facilities, but it was quite a paradise to me. And I went there to study primate behavior. I was an undergraduate in anthropology. I compared macaques and gibbons and how they move through the trees. This national park is a refuge for many lowland animals like orangutans, many of the wild populations there. It's had very little hunting historically. So it's a very important place in terms of biodiversity conservation. You can see if you kind of remove it there, it's a long ways to any other habitat that might be appropriate for these animals. The park was surrounded by logging operations at that time. This is 20 years ago. But unfortunately, a lot of it's been converted to oil palm plantations by now. And so working through the forestry department, we approached the logging company that was just on the north side of the park and approached them about studying their forests. And they were quite cooperative, and they allowed me and a couple of assistants to set up camps in various places. And we conducted large-scale surveys. We had 20 transects just to survey forest condition. And so we would go and set these things up in three different forest types. This is a raw data sheet here showing you kind of how I had the forest conditions marked as we did the transects. This is from the first publication showing you actually the layout of the transects and where the plots were as well. So while we did the transects, we were also setting up plots that were kind of stratified according to the forest condition that we saw out there. These are kind of indicating the different types of forest plots that are on that figure with logged and unlogged forests. And this is where the morphospecies concept comes into play, because in the plots we measured and studied all of the trees. One part I took detailed field descriptions of every stem in these plots, and this is a sheet that I filled out for every tree that I encountered. It kind of has descriptions of all the aspects of the tree, the outer bark, as you can see here, it's circled in more detail, and I had a system for marking the primary and secondary features, and so made quite detailed descriptions of all of these stems. And so in thinking about this study, how can we bring this citizen science into this critical key aspect of the morphospecies? So I made fallen leaf collections for all of these things. I have them organized. They're pressed, and here they are in envelopes in a cabinet organized by family. And they also have species numbers here. And so here's the whole collection there you can see on the left side coming from that study. And kind of just for fun, I'd like to show you how I did this morphotyping. I create these key rings. This is the Lesithidaceae family. These are samples from that family. I'd put them in plastic bags and label them, and I'd keep them all together on a wall with all the other families. And so I had these rings with the sample numbers and with the species identification number. And so every unique morphotype that I found would go up there on the wall and be incorporated into this key ring. And so this is a very useful thing. I had a whole wall full of these things for all the families in my study. And it was quite an effective way to do the morphotyping. Each unique morphotype went up on the wall as I progressed. In this study, we compared the tree diversity between undisturbed forest and selectively logged forest nearby. And we came up with a rather controversial result at the time that the selectively logged forest was actually pretty diverse. In just 8 years, we saw a significant recovery of the tree diversity in these forests. And this wasn't just a recruitment of pioneer species. Many of these species were present primarily in the undisturbed forests as well. So you can see this morphospecies concept is very central to this study. So I think this study would be a perfect test for looking at whether or not we can bring citizen science approaches into biodiversity studies, where expertise is often a major shortcoming. We just don't have enough of it. So if we can develop a way so that citizen scientists can contribute in a meaningful way and actually maybe improve our rigor by giving us estimates of variance and so forth, I think this would be a very useful thing for us to be able to exploit. So Elva and I will be taking standardized photos of all the fallen leaf samples that I have from that study. And we'll be making these available, and we'll begin to explore whether this actually gives us meaningful results and whether it actually gives us more power in understanding the morphospecies concept.
And I'd like to address some of the questions the students had that weren't so familiar with the morphospecies concept. It really is primarily an ecological tool that's been used in many studies across the world, really, to identify how many species are in an ecological community and to compare that across a landscape or even a continent. So instead of providing you with what I think are the answers, I'd actually like to leave you with a number of questions. First is, how does a taxonomic species differ from a morphospecies? So you have keys and things, you have a long list of characters that you might describe a taxonomic species, but really how does it differ than simply looking at them and distinguishing them based upon their morphology?
At what geographic scale would you think most morphospecies would be useful? Certainly within a single river valley or watershed, you could compare the individual trees there and decide how many species there are. What about if you went up to the entire island, the entire continent? Could you really make a useful morphospecies at that geographic scale? Given your particular organism of interest, what do you recognize as the major limitations in our understanding of biodiversity? Is it simply collecting? Do we simply need to get out there a lot more and collect many different areas? Or is it actually detail? Do we need a lot more detail in understanding how biodiversity varies? What is the major limiting factor in our understanding of biodiversity right now? So I'll leave you with that for right now, and we'll soon have a lecture about metagenomics and species barcoding. These advanced techniques might be able to provide us with the kind of detail and the automation that could really move biodiversity research forward very rapidly. But again, I don't think we should totally abandon this morphospecies concept, and we should continue to explore how we might be able to adapt it and modernize it. See you again soon.